Hello, this is Fabrice Gorek from the macromolecular crystallization facility at the Laboratory of Molecular Biology in Cambridge, UK. The facility has about 80 users, mainly from our structural studies and PNSA departments. We also welcome scientists from the University of Cambridge. As part of the IUCR's webinar program, I'm going to talk about the formulation of the MORPHEUS protein crystallization screens developed within our facility. Technology developments, including innovative screens, are needed to obtain X-ray diffraction quality crystals from increasingly challenging protein samples. Crystallization screens are continuously developed at the LMB, essentially to further enhance the yield of our initial leads. MORPHEUS screens integrate small molecules frequently observed in the PDB to co-crystallize with proteins. In that sense, they are PDB-derived ligands. These molecules are employed mostly as additives that act as protein stabilizers, crystal packing bridges, or any other roles beneficial to protein crystallization. Each MORPHEUS screen integrates 96 conditions, a minimal format adapted to automation and limited amount of sample. To minimize the number of conditions employed, the molecules found in the PDB are combined into mixes of additives. Other mixes of compounds have been developed, such as precipitant mixes that also act as convenient and effective cryoprotectants, and also buffer systems that facilitate the pH optimization. The different types of mixes are combined using a fixed ratio to generate 3D grid screens. The preparation of a MORPHEUS screen and the optimization of its conditions are amenable to automation. Before making final choices about the formulation, a multitude of test screens are used against challenging samples produced at the LMB. The main goal is to produce exclusive hits that are useful for solving novel structures. Before describing the developments related to the MORPHEUS screens, I will briefly present some aspects of protein structure determination by X-ray crystallography, including of course matters related to crystallization and crystals. Theoretical and experimental aspects were taken into consideration during the early stage of the development. This to introduce screens particularly adapted to the current needs in the field of X-ray crystallography. MORPHEUS screens 1 and 2 are now used routinely in many laboratories, while MORPHEUS 3 is still being developed at the LMB. We show examples of optimization of a MORPHEUS condition following the four-corner method in a complementary webinar. Major breakthroughs about protein structure determination start in the 60s. Crystallographers like Kendrew and Perutz were then using globular proteins that were abundantly available, such as hemoglobin. Hemoglobin readily crystallizes in the shape of large and robust crystals. High-quality diffracting crystals of hemoglobin were obtained in vials containing few milliliters of saturated ammonium sulfate or phosphate solution. The limiting step then was essentially about processing the diffraction data. With limited computer power, calculations were taking years. The resulting models were crafted, such as the one shown here on the right, made in the LMB workshop. The rate of structure determination increased dramatically in the last 30 years, notably because of advances in molecular biology, IT, and software developments, and also the introduction of robotics for high-throughput crystallization. The PDB now holds over 120,000 coordinate sets, a vast majority of which resulted from X-ray crystallography. This is shown on the graph here, with rates of structure deposition in the PDB according to different techniques. The rate of structure determination by using X-ray crystallography has however slowed down in recent years. See the purple plot. An underlying reason is the increasingly challenging nature of the samples investigated. Some samples contain fibrous, disordered proteins, and also their complexes. Meanwhile, electron microscopy is gaining popularity as it enables structure determination with very large complexes. Using recombinant DNA technology and protein engineering, Crystallographers spend now a lot of time trying to obtain samples of interest that can be crystallized. This histogram, produced by the group of Naomi Cheyenne at Imperial College, results from compiling data of structural genomics. We can calculate that 18% of soluble samples come from initial clones, and about 1 out of 10 soluble samples will later enable structure determination. Despite all the efforts to optimize the samples, the yield of quality diffraction crystals is often very poor. In fact, we regularly observe a percentage of useful hits below 1% during initial crystallization experiments at the LMB, as for the examples shown in the table here. Again, an underlying reason is the increasingly challenging nature of the samples investigated. Crystals we eventually obtain are typically unstable and fragile. Mostly hydrophobic interactions hold a crystal together, and the solvent content is high. Besides, crystals are relatively small. These characteristics makes the handling of crystals difficult. Finally, high mosaicity is usually observed later. Crystals are cooled down to cryogenic temperature by flash cooling, essentially to reduce radiation damage during data collection. Prior to flash cooling, the crystal must be soaked in the condition that is now cryoprotected, and hence diffraction is not lost because of the formation of ice. To cryoprotect a condition, glycerol is usually integrated into the condition until vitrification is observed after flash cooling. However, crystal quality can be lost also because of the late integration of a reagent in the condition, and the additional handling of crystals required for soaking experiments. After data collection, the phase problem often means failure to solve novel structure, since there are no known homologues to apply the molecular replacement method. An experimental method to solve the phase problem is based on anomalous diffraction. Insertion of a heavy atom into crystals is typically required. This can be done with soaking crystals in the condition where a heavy atom has been added, a step called heavy atom derivatization. However, heavy atoms are large and hydrophobic, and hence soaking assays usually result in loss of crystal quality. Finally, different heavy atoms should be tested in order to find the one that binds specifically with minimum diffraction loss. Crystallization is a stochastic process which requires empirical approaches. There are several methods employed for screening crystallization conditions, with essentially batch crystallization, dialysis, free interface diffusion, and vapor diffusion. Vapor diffusion is a widespread method. It is efficient and amenable to automation. Here is represented a cross-section of a sealed crystallization chamber during vapor diffusion. The reservoir is filled with a condition, and the upper wells contains a droplet composed from both the protein sample and the condition. Because normally the precipitant in the droplet is less concentrated than in the condition, the concentrations of all the components in the drop rise through water lost during equilibration by vapor diffusion. This is what a phase diagram generally looks like for globular proteins. The pathway to crystallization is here shown for different methods. A phase diagram is based on two parameters, typically the protein and precipitant concentrations. The goal of any crystallization experiment is to reach a supersaturation state where the system undergoes internal changes and nucleation eventually occurs. It is important to understand a phase diagram is unavoidably incomplete, considering the multitude of other parameters to be investigated, such as the pH or temperature. In addition, a protein can exhibit much more complex behavior during crystallization. We show here a less typical phase diagram produced after investigation on clarthrin. There are three main categories of crystallization reagents: precipitants, buffers, and additives. Although some reagents have physico-chemical properties that make them well suited for different categories. Ultimately, a precipitant is found at relatively high concentration, basically close to saturation, while buffers and additives are found at relatively low concentration, 0.2 molar or lower. Main precipitants are small salts, like ammonium sulfate, organic solvents, like ethanol, and polymers, with notably polyethylene glycols, called PEGs. Precipitants strongly attract water molecules and trigger salting out. When the precipitant concentration is increased, there are less water molecules available to interact with the charged surface of the protein. As a result, protein molecules form hydrophobic interactions with each other. Eventually, these interactions lead to crystallization. Other parameters than salting out are altered according to the nature of the precipitant, such as the dielectric constant of the condition or the kinetic of the experiment. PEGs are widespread crystallization reagents. As polymers, their length is directly correlated to their molecular weight, and so are their physicochemical properties. PEGs not only compete with proteins for water, they accumulate interesting properties. Notably, the smallest PEGs are well known to be cryoprotectants. The density of charges on the surface of a protein varies according to pH. In other words, the different buffers found in crystallization screens alter the protein solubility and possibilities of protein-protein interactions. Biological buffers, such as HIPs and MEs, are widely used because they don't affect the stability of most proteins. Other common buffers are small carboxylic acids. Additives can alter the parameters related to crystallization in multiple ways. They can increase the chances of obtaining useful crystals simply by tuning the effects of the other reagents. They can also stabilize the protein, or alter its surface landscape and structure. Additives that alter the stability and solubility of a protein sample can be selected prior to crystallization with a thermal Schiff assay. However, other kinds of positive impacts from additives cannot be anticipated, such as when an additive enables crystallization by cross-linking, or later enables experimental phasing. Useful additives include molecules with a multitude of possible binding modes via different spatial arrangements of functional groups. Here we show Fischer projections for 4 monosaccharides and their hydroxyl groups. Sugars are well known for their thermodynamic stabilization of macromolecules. Many sugars are often seen ordered in crystal structures deposited in the PDB, although they are not frequently integrated in initial conditions. Additives can mediate crystal contacts that helps to form the crystal lattice, like it has been observed when using multivalent metal ions. An example is shown here with divalent metal ions that bound the his tags of a protein and induced a trimer. This trimer was not observed in cells, but was essential to obtain quality diffraction crystals. The combinations of reagents alter the combinations of variables associated with the main parameters of crystallization. There are different approaches to initial screening according to the amount of sample available and the type of protein investigated. A 3D grid screen consists in a systematic approach to screen formulation. The small cubes shown here represent 48 combinations of 2 types of reagents and pH values in a conceptual space. Because of the very large number of reagents well suited for crystallization, an extensive grid screen will include millions of combinations. This is technically not feasible, notably because of the limiting amount of sample. Subsequently, most developments of initial screen are based upon minimizing the number of initial conditions. Historically, there have been 2 main methods to formulate minimal screens, one based on incomplete factorial, the other called sparse matrix. Carter and Carter suggested the incomplete factorial method for crystallization in 1979 by randomly assigning combinations of factors to individual experiments and later formulate the most adapted screen for their protein. Here is represented an incomplete factorial with 48 conditions. And here is represented a sparse matrix, still with 48 conditions. A sparse matrix is an empirical selection of conditions which are known to have crystallized a subset of proteins. An approach originally suggested by Jean Carrick and Kim in 1991. Ideally, minimal screens should be formulated routinely and biased towards the characteristics of the sample investigated. However, the technical requirements for producing routinely various types of conditions and screens from a multitude of reagents are challenging. In fact, most crystallization facilities purchase standard minimal screening kits. More or less kits are combined to form a routine initial screen. Automation enabled high-throughput crystallization. An important aspect of our strategy at the LMB is to proceed with a relatively large number of initial conditions when the amount of sample allows it. Users of a facility are being given the opportunity to screen their samples against over 2,000 conditions. For this, various standard minimal screening kits are assembled into pre-filled MRC96 well plates by the fully automated system shown here. Even when employing a relatively large number of conditions, our initial screen is not efficient enough, because many samples fail to crystallize or give only a very few hits. In 2013, when plotting the number of times reagents appear in our initial screen against the number of times they are seen in our published conditions, a correlation was observed. I argued that an underlying reason is the undersampling of reagents. In other words, since crystallization is a stochastic process, increasing the variety of reagents and conditions should increase the chances of obtaining quality diffraction crystals. Considerations we've seen so far were taken into account to formulate the MORPHEUS screens, starting with the original screen, MORPHEUS 1. The corresponding work was published in the Journal of Applied Crystallography in 2009. The name Morpheus comes from the Greek god of dreams, who was associated with premonition. Morpheus was taking different forms in people's dreams to convey a message from the gods. Crystallographers often dream that crystals take different forms. So what are the main ideas behind Morpheus formulation? Well, each condition integrates more reagents than traditionally, with a mix of additives, a mix of precipitants, and a buffer system. Note that cryoprotectants are part of the precipitant mixes, since generally they are also precipitants. The multiplexing of reagents has been performed previously. I invite you to read the publications cited here that inspired Morpheus formulation. The multiplexing of reagents was applied to virtually expand a 3D grid screen. Another idea was to integrate undersampled reagents and mixes that are well suited for crystallization. Our large initial screen was an ideal source of information for that matter. For example, we observed some mix of precipitants were undersampled but efficient. Also, by selecting components often seen ordered in crystal structures of the PDB, the chances of incorporating molecules playing a positive role should increase. In this perspective, not only undersampled reagents should be integrated. By formulating a screen from scratch and base its development on a broad variety of samples tested, bias should be minimized. Finally, demands of optimizations, cryocrystallography, and solutions to the phase problem should be taken into account more. Ideally, some steps along the process of structure determination should be bypassed. This is why cryoprotectants and heavy atoms were integrated. Small molecules and ions found abundantly in the PDB are potentially good crystallization agents. Surely, as additives, they can make proteins behave differently. For Morpheus 1, PDB-derived ligands that are not larger than 250 daltons, well-soluble, stable, and readily available were selected. While small and soluble, the chances of unfavorable interactions that would prevent them from binding efficiently or destabilize the protein should be minimized. 38 PDB-derived ligands have been grouped into families depending on their chemical nature to formulate the mixes, including 8 additive mixes, 4 precipitant mixes, and 3 buffer systems. The 3 types of mixes forming each MORPHEUS condition are combined using a fixed ratio. The resulting 3D grid screen is represented here with a schematic layout. An example of additive mix is shown here with the formulation of the corresponding stock. This mix integrates 6 sugar derivatives. By grouping additives with similar chemical nature, risk of cross reactions are reduced, and hence the corresponding mixes are relatively stable. It is important to understand the concentration of a mix is the sum of the concentrations for each reagent. In this case, 6 times 0.2 molar equal 1.2 molar. Also, the concentration of additives in the stock is 10 times the final concentration in the screen. The table shows the formulation of the 4 stocks for the precipitant mixes. 3 of the mixes have been observed to be more successful than expected in the crystallization of LMB samples when considering they were undersampled in our initial screen. A 4th mix was designed from scratch with components not found in the other 3 mixes. The final concentration of a cryoprotectant was set to produce vitrified conditions during flash cooling. The cryo used in MORPHEUS 1 were already well known from crystallographers. Less known reagents were employed in MORPHEUS 2 and will be presented later. As for the stocks of additive mixes, the concentration of a stock is a sum of concentrations. However, the concentration of precipitants in the stock is now 2 times the final concentration in the screen. 6 of the selected PDB-derived ligands have been used to build 3 buffer systems with pH 6.5, 7.5, and 8.5. Each MORPHEUS buffer system includes an acid and base pair of buffers with close pK values. This way, a system combines the characteristic of 2 different biological buffers. Non-titrated stock solutions of the individual buffers can be mixed at different ratios for optimization purposes. Recipes for preparing 50 ml of the buffer system imidazole are shown here, starting with concentration of 1 molar. The concentration of the buffers in the stock is 10 times their final concentration in the screen. Alex Berndt obtained initial crystals of a kinase catalytic subunit exclusively in a test screen during the development of MORPHEUS. This kind of early results motivates other researchers to try the test screens and drives the final choices of formulation. According to a recent query in the crystal growth procedures from the PDB, which is vastly unreported, MORPHEUS is mentioned over 200 times. Here, and on the next slide, examples of structures solved using the MORPHEUS screen are shown. Although a very large structure is usually the result from combining X-ray crystallography and cryo-EM techniques. As anticipated, the screen is suitable for a broad variety of samples, including proteins that tend to polymerize, large proteins, complexes, viral proteins, and others. See on the right an example of transmembrane protein that crystallized in MORPHEUS. MORPHEUS 2 was formulated in continuity with MORPHEUS 1. The corresponding work was published in ACTA F in 2015 as proceedings of the 15th International Conference on the Crystallization of Biological Macromolecules, held in Hamburg. MORPHEUS 2 follows the formulation principles and developments of the original MORPHEUS. However, different reagents were investigated, with notably metals that are amendable to the collection of anomalous datasets. Non-detergent sulfobetaines and polyamines, which are known to enhance the solubility and stability of many proteins. Most chemicals were still selected as small PDB-derived ligands, although few reagents that were not found in the PDB were integrated to complete the formulation, with essentially 4 glycerol-like cryoprotectants. Finally, more innovative buffer systems were integrated to the formulation. Here is the formulation of the stock solutions for the 8 additive mixes of MORPHEUS 2. Like in MORPHEUS 1, the first mix is composed of very common salts. Several mixes were prepared with metals, such as lanthanides. 2 other mixes integrate monosaccharides and amino acids. There are different sets from MORPHEUS 1. Finally, the polyamines form the 8th mix. We assume the role of NDSBs was less specific, and therefore they have been integrated to the precipitant mixes instead. An investigation to formulate MORPHEUS 2 was testing almost 100 commercially available polyols for cryoprotection properties. On the left, we can see a list of some of the chemicals tested. Initially, each polyol was prepared at different concentrations, and vitrification of the corresponding solutions by flash cooling was assessed by eye. Cryoprotection was later investigated on an X-ray diffractometer for fewer polyols. It was found that 4 small polyols that were not currently found in any commercially available screens were as efficient as glycerol. These 4 cryoprotectants are shown on the right underneath glycerol. Andrei Sheshak obtained one of the first exclusive hits in a test screen during the development of MORPHEUS 2. Crystals of a bacterial homologue of actin that integrated heavy atoms grew in a condition with lanthanides. Significant anomalous signal was induced that enabled structure determination without further optimizations. The development of crystallization screens from scratch, regardless which approach to formulation, is mostly empirical. A major difficulty for a screen like MORPHEUS is the relative complexity of the conditions, that include up to 13 reagents. The solubility of the reagents need to be tested initially. Some may not be soluble enough within the pH range selected, or incompatible with other chemicals when mixed. The mixes and conditions are later tested for crystallization with proteins that crystallize readily. At this stage, we basically check that vapor diffusion and salting not occur, and also if additives are suitable for crystallization. Unfortunately, some useful heavy atoms could not be integrated, such as those of the platinum group. They could not be solubilized and are very unstable in solution. Other chemicals were discarded because of incompatibility and false positives. Test screens are made available to the users of our crystallization facility, along with many commercial screens. When there are no interesting results with a type of mix or condition, the subsequent test screen will integrate different ones. The conditions must be relatively stable, so the screen can be stored for a few months in MRC plates. Although the ultimate goal is efficiency, and compromises on stability may be required. For example, the conditions integrating lanthanides at pH 8.5 tend to form light precipitates, but were useful, and hence they are part of the final formulation of MORPHEUS 2. When several exclusive crystallization hits have been obtained, final choices are made about the formulation. When users clearly appreciate the usefulness of conditions already cryoprotected, it has not been possible to evaluate the impact of a broader variety of cryoprotectants on her structure determination process. However, I would argue that formulation should not be biased towards only glycerol as main cryoprotectant, because this will contribute to undersampling. Basically, more convenient and efficient cryoprotectants should be integrated in initial screens in order to alter parameters associated with flash cooling. The concentration of additives is set relatively high because low affinities should be considered. In addition, although the additives were selected as potential ligands, some may have other useful roles that will not be observed at very low concentration, such as tuning the effects of a precipitant, for example. The relatively high concentration limits the number of additives that can be integrated into a mix. Ultimately, the availability of reagents is also a criteria. Rare molecules can only be investigated at later stage, when more structural data is available for the sample investigated. A screen with a number of conditions limited to 96 is unavoidably incomplete. Since the concentration of MORPHEUS precipitants had to be fixed, a relatively high concentration was set, as it would give more chances to observe hits when the starting concentration of a protein is quite low. Some proteins may require harsher conditions to crystallize, such as conditions saturated in amino sulfate, or with extreme pH. That's why, when attempting crystallization with a novel sample, I would argue that a multitude of screening kits based on different formulation approaches should be employed when possible. Ideally, each type of formulation should not be minimal, but extended to hundreds of conditions. Next, we are going to introduce some details about the development of MORPHEUS 3, where more reagents are being investigated. MORPHEUS 3 still follows the formulation principles and developments of the original MORPHEUS. However, the selection of additives cannot be any more restricted to high occurrence in the PDB and a molecular weight below 250 daltons. Choices of relatively soluble and readily available compounds, according to these original criteria, is narrowing. We are investigating the integration of drug compounds, such as peptides, vitamins, and cholic acid derivatives. So there will be possible applications to drug discovery with MORPHEUS 3. However, the main goal stays the production of exclusive hits useful for structure determination. Well, these developments would not have been possible without the hard work and enthusiasm of scientists and students from the LMB. And I thank the group of Ian Lowe, to start with, the group of Roger William, and Crystallization group. I also thank our visual aids department, notably Joe, Matthew, and Neil, people from the International Union of Crystallography. One of the IUCR many missions is to build on outreach and capacity building in all geographical locations. I must state a conflicting commercial interest, since MRC technology commercializes all products derived from MORPHEUS formulation under an exclusive license to molecular dimensions. And I'm here with Janet Hobbs, product manager at Molecular Dimensions, who works on the production and commercialization of the MORPHEUS screens, so that many other labs can access our technology.